Researchers around the world are a step closer to a better understanding of the intricacies of COVID-19 thanks to two new web resources developed by investigators at Baylor College of Medicine and the University of California, San Diego. The resources are freely available through the Signaling Pathways Project, Baylor, and the Network Data Exchange, UCSD. They put at researchers' fingertips information about cellular genes whose expression is affected by coronavirus infection and place these data points in the context of the complex network of host molecular signaling pathways. Using this resource has the potential to accelerate the development of novel therapeutic strategies. The study appears in the journal Scientific Data. Our motivation for developing this resource is to contribute to making research about COVID-19 more accessible to the scientific community. When researchers have open access to each other's work, discoveries move forward more efficiently, said leading author Dr. Neil McKenna, Associate Professor of Molecular and Cellular Biology and member of the Dan L. Duncan Comprehensive Cancer Center at Baylor. However, usually this information is not easily accessible. In the current study, the researchers generated consensus for genes affected by infection with three major coronaviruses: Middle East Respiratory Syndrome Coronavirus (MERS) and Severe Acute Respiratory Syndrome Coronaviruses 1, SARS-1, and 2, SARS-2, which causes COVID-19. The resource identifies the genes whose expression is most consistently affected by the infection and integrates those responses with data about the cell's molecular signaling pathways, in a sense getting a better picture of what happens inside a cell infected by coronavirus and how the cell responds. The collaboration with UCSD makes our analyses available as intuitive cytoscape-style networks, says McKenna. For example, the connection between pregnancy and susceptibility to COVID-19 has been difficult to evaluate due to lack of clinical data, but McKenna and colleagues' approach has provided new insights into this puzzle. This finding suggests the hypothesis that the suppression of the interferon response to SARS-2 infection by elevated circulating progesterone during pregnancy may contribute to the asymptomatic clinical course, McKenna said. Consistent with their hypothesis, while this paper was being reviewed, a clinical trial was launched to evaluate progesterone as a treatment for COVID-19 in men. Scott A. Oxner at Baylor College of Medicine and Rudolph T. Pilich at the University of California, San Diego were also authors of this work. This study was supported by the National Institute of Diabetes, Digestive and Kidney Diseases NIDDK Information Network DK097748, the National Cancer Institute CA125123, CA184427, and by the Brockman Medical Research Foundation. The Signaling Pathways Project website is hosted by the Dan L. Duncan Comprehensive Cancer Center. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.